Hello students. Today we are going to see about DNA replication process. I think you all know that in molecular biology, DNA replication is very important biological process of producing two identical copies of DNA from one original DNA molecule. And it's a biological polymerization reaction, so which proceeds with the sequence of initiation, elongation, and termination. So this is a full and full enzyme dependent reaction. Okay, along with the DNA polymerase, there are different enzymes involved in the DNA replication process. So before the DNA replication process, you must know what is the need of DNA replication. Okay, all the cells must replicate their DNA before the cell division, right? So this makes the each new cell produced during the cell division. It must receive the all the genetic material necessary to survive and reproduce. So this is very important biological process before the transcription and translation. And one more thing you must know that the DNA replication is a semi-conservative method. Okay, why we are calling this DNA replication is semi-conservative means, so each DNA strand serves as a template for the synthesis of the new strand and producing two new DNA molecules, each with one new strand and one old strand. Okay, so during the DNA replication, the double-stranded DNA will denature and one strand will serve as a template and one strand is old and the synthesizing strand is new. So one is new and another one is old strand. So that's why we are calling the calling DNA replication as a semi-conservative replication. I think you know that the enzyme involved in DNA replication in prokaryotic cell and eukaryotic cell. We have already seen in detail about the DNA polymerase, different types of DNA polymerase in prokaryotes and polymerases in eukaryotes. I think you know, right? DNA polymerase 3 is involved in the prokaryotic DNA replication, and DNA polymerase delta is involved in the replication in eukaryotic cell. Now we'll see in detail about the DNA replication process. So before the DNA replication process, you must know what are the enzymes involved in the DNA replication. Okay, I have already told that this DNA replication is a full and full enzyme dependent mechanism. Okay, so the DNA replication would not occur without the enzyme. In this slide, you can see the various enzymes involved in the DNA replication. Okay, for example, and also you must know that DNA polymerase is the key enzyme is needed for the DNA replication. You know that, but DNA polymerase can't initiate the replication process. So it's depend upon the some other enzyme to unwind the DNA, like DNA helicase for unwinding the DNA and DNA primase, like. So it's depend upon the other enzyme to start with the DNA replication. Okay. So in this picture, you can see the mechanism of DNA replication and the whole mechanism of DNA replication. We'll see one by one. The steps in all DNA replication is initiation, elongation, termination. The first step is formation of replication fork. Okay, so the before the DNA can be replicated, the double-stranded DNA molecule must be broken or unzipped into two single strands. Okay, so the double-stranded DNA must be unwind. So for this, the cell need the DNA helicase enzyme. So what this helicase will do means this helicase will simply it will unwind the DNA. How it will unwind the DNA means it will break the hydrogen bonds between the base pairs. You know, right? The bases are linked by hydrogen bonds. So this DNA helicates, it will disturb the hydrogen bonding between the base pair and it will separate the DNA into Y shape. So Y shape. So now you can after the DNA helicase enzyme action, the double-stranded DNA will be break and it will look like a Y shape, and we are calling as a replication fork. Okay, now the one strand it act as a template for the replication to begin. Now you must think the orientation of the DNA. One strand is 3 prime to 5 prime, another strand is 5 prime to 3 prime. Both the strands are act as a template for DNA replication process. Okay, the next step is primer binding. Okay, once the DNA strand have been separated, the a short piece of RNA called primer, it will binds to the 3 prime end of the strand. So the primer is nothing but this is a small nucleotides will be attached by the enzyme called DNA primase. Okay, so the DNA polymerase it can't initiate the replication, so it depends upon the helicase and primase enzyme. So it's DNA polymerase it won't directly add the DNA nucleotides. For the binding and for startup of the replication process, the DNA primase enzyme is very important, and you must note that here the DNA primase it won't add the DNA nucleotides, it will add only the RNA nucleotides just for the startup. Okay, next is elongation. Here only our DNA polymerase 3 will come and play. Okay, so in prokaryotic cell, there are different DNA polymerases. One is DNA polymerase 3. So this is the main enzyme involved in the synthesis of the new DNA strand by adding the DNA nucleotides, and other enzymes also there like DNA polymerase 1, 2, 4, and 5. These enzymes are responsible for error checking and repair mechanism. So what this DNA polymerase 3 will do? It will bind to the strand near to the site of the primer. Okay, the primase added the primer, right? So it will go near to the attached to the primase enzyme. It will be start adding the new complementary DNA nucleotides. Likewise, in eukaryotic cells, DNA polymerase delta do the same process. So since the DNA replication proceeds in the 5 prime to 3 prime direction and the leading strand, and the newly formed strand is continuous. Okay, so in the leading strand, the strand synthesis is continuous, but in the lagging strand, the DNA replication is by binding with multiple primers. Here, the same mechanism in leading strand, only one primer will is produced and produced by primase enzyme, and the further elongation will be takes place by DNA polymerase 3. But in the opposite strand, the multiple primers will be added by the primase enzyme, and the multiple DNA polymerase will come and produce different multiple segments. Okay, these different multiple copies of small fragments are called as Okazaki fragments. This is a discontinuous process. In the lagging strand, the DNA replication is a discontinuous process, but in case of the leading strand, the replication is a continuous process. Okay, you just in the parallel side, you just see the picture. Okay, then you can understand the process very easily. So the last step is termination. Once both the continuous and discontinuous strands are formed, an enzyme called exonuclease, it will remove the RNA primers. Okay, it is a DNA replication. So the final copies of DNA must having the DNA, not the RNA. So the RNA primers must be removed by exonuclease enzyme. Automatically, these the RNA base pairs are replaced with the appropriate DNA bases. Now, another exonuclease, it will proofread the newly formed DNA. Proofread means what? It will go and check the continuous full DNA, and it will check whether the DNA nucleotides are added correctly or not. So all the mismatch base pairs will be removed by the exonuclease enzyme, and it will continuously synthesize the DNA. Another enzyme called DNA ligase, it will join the Okazaki fragments. Finally, the single length of the DNA strand will be produced. Once the process is completed, the parent strand and its complement strand is coils into the familiar double helical shape. In the end of the replication, produce the two DNA molecule, each with one strand from the parent molecule and one strand from the newly synthesized DNA. So in this slide, you can see the difference between the prokaryotic and eukaryotic replication. In the prokaryotic cell, the replication is occurs inside the cytoplasm, but in case of eukaryotic cell, it occurs inside the nucleus. Only one origin of replication is present in per the DNA molecule, but here in eukaryotic cell, there are many origin of replication. The speed of the replication is 100 to 200 nucleotides in length, but here in eukaryotic cells, in 150 nucleotides. In the chromosome of the prokaryote, the replication occurs at only one point, but in case of the eukaryotic cell, replication occurs at several points simultaneously in each chromosome. Okay, only one replication fork is formed because of the one origin of replication, but in case of eukaryotic DNA replication, there are multiple replication forks are formed. Then there are some other proteins are also involved in the initiation of the prokaryotic DNA replication, DNA A and DNA B. In case of the eukaryotic cell, the initiation is carried out by origin recognition complex. And also to DNA guidase or topoisomerase needed for the prokaryotic DNA replication. For eukaryotic DNA replication, DNA guidase is not needed.